All right, folks, here we go with our lesson 6, carbon-14 dating. I'll get you started with a little video clip here.
Suppose you've discovered an ancient bone amulet in an archaeological dig and you want to know its age. You can use the rate of decay of carbon-14 to determine the artifact's age. The nucleus of a carbon-14 atom is unstable. At some time, this unstable nucleus will emit a high-energy electron called a beta particle, which has a 1- charge. The beta particle causes a neutron to change to a proton, and the carbon-14 atom then decays into a nitrogen-14 atom. The average time for half of a group of carbon-14 atoms to decay is 5,730 years and is called a half-life. Your bone amulet is found to contain only 12.4% of carbon-14. How old is the artifact? The level of 12.4% indicates that 3 half-lives have passed. 3 half-lives times 5,730 years is 17,190 years old. Carbon-14 samples can be accurately dated up to 60,000 years old.
All right, so there we go. There's a little intro to carbon-14 dating. Now let's look a little deeper. All right, so the definition for our carbon-14 is, as you may have caught from the video, is it's the amount of time it takes for a sample of carbon to lose half of its radioactive C14. Hence the name half-life. Remember, carbon-14 is radioactive, which means it's unstable, so it breaks down over time. This little animation kind of shows you what's taking place here. So over time, the yellow spheres, which represent carbon atoms, are turning into nitrogen atoms because the one of the neutrons shoots off of a electron and becomes a proton. So now the yellow spheres represent the carbon and the blue spheres represent the nitrogen. So how does it work? In a sample of carbon, the radioactive C14 will decay or break apart at a specific rate, and we don't really know why it's the rate that it is, but we do know what the rate is. So this is sort of my connection to our activating activity, where you're looking at the picture of the dead pig. All right, so the basic idea here is if you had, say, 100 or so C14 atoms, then in that half-life of 5,730 years, which you might want to jot down on your paper somewhere, wherever you can find a spot, you can see that half of them have turned into nitrogen atoms and the other half still remain carbon-14. So, in living things and in the atmosphere, the ratio is about the same. About 1 in a trillion carbon atoms are C14, and it doesn't do anything to us when we have it in our bodies. They're just there, and they eventually break down over time. The ratio stays the same or constant as long as you stay alive. By eating food, you're gaining as much C14 as you're losing. So our pups here they're keeping the C14 at the same rate. When something dies, though, it begins losing its C14 at the constant half-life rate. And of course, since it died, it is no longer eating. Therefore, when it loses the C14 that it had, it is going to not have as much anymore because it's not eating anything, so it's not taking in more carbon-14. Here's a way to kind of think about it visually. So you have your plant, okay, and the bear eats the plant. So we don't know exactly how many carbon-14 atoms are in this bear, but we do know the ratio. So when he dies, he's no longer eating, and his body begins to lose the C14 at a constant rate. So after 5,730 years, he's only going to have half of the carbon-14 atoms that he had initially. when he was alive. So, moral of the story is by measuring how much C14 is left, we can estimate how long ago that thing died. So, if you think of a tree, it's going to have it takes in carbon from the atmosphere in the form of carbon dioxide, and it grows into the tree. Then after one half-life, 5,730 years, it's going to decay, and it's only going to have 50% of the C14 left. If you go another half-life to 11,460 years, then it's only going to have a quarter, 25%, of the original amount of carbon-14 that it had. And then after another half-life, it's going to be that 25% will become 12.5%, and you would see even smaller amount of C14 in that. So let's take a look at how scientists do measure the C14.
How do we know how old something is? For people, we'd ask to see their birth certificate. For trees, we'd count the rings. But how do we know how old a fossil is? Fossils have their own internal clock. Scientists can read it by looking at the ratio of two different types of carbon atoms. Of course, every living thing is made of carbon. Plants grab carbon dioxide from the atmosphere and use it to form complex organic molecules. Animals get their carbon by eating these plants. But there's more than one form of carbon. Most carbon atoms have 6 protons and 6 neutrons. We call this carbon-12. High up in the atmosphere, sometimes cosmic rays hit nitrogen atoms. This creates carbon with 6 protons and 8 neutrons. We call this carbon-14. Carbon-12 and carbon-14 behave alike, but carbon-14 has one unique and important attribute. It's unstable. So once an animal dies, the carbon-14 in its body will start to go away. Every 5,730 years on average, about half of the carbon-14 atoms will decay into nitrogen. This is its half-life. After one half-life, the animal will have about half the amount of carbon-14 it started with. After another half-life, it will have about a quarter. And after another half-life, it will have about an eighth. By contrast, the amount of carbon-12 it has in its body will stay the same. By measuring the ratio of carbon-14 to carbon-12, we can measure how many thousands of years have passed since the animal died. Carbon dating works for fossils up to about 60,000 years old. For older fossils, scientists use unstable elements that have much longer half-lives. For Scientific American's Instant Egghead, I'm Michael Moyer.
So there you go. That's how scientists measure the amount of C14 left in a dead thing. Right? And you're actually getting ready to do a little of that on your own here. So we'll move on to our lab directions for our carbon-14 activity.